this is Roger Green, host of the SurfingMASH Tsunami podcast. This week, we're offering 4 conversations from episode 19, our interviews with Mike Patel, Louise Campbell, and Sven Franke about their impressions of the EASL Congress meeting. Plus, instead of a vault, a special excerpt we cut from Louise's interview so that we could get the overall episode down to an hour and 10 minutes. It's got its own content, its own topic, and it's pretty powerful in its own right. This conversation with Louise took place Saturday afternoon after she had flown back to London. It focuses largely on Friday sessions with some comments from me about the late breakers and some chat between the two of us about the implications of what we've been seeing. We cover an array of topics in that time, including her general sense of the meeting, the importance of the new CPG guidelines, which we will be discussing more fully in episode 21, posted on June 27th, which Louise finds extremely exciting, and then a discussion of some of the drugs covered in the light breaker and general sessions. I asked a question about the relative merits of CAP versus PDFF as an in-office measure, which evolved into a question around data quality and what a competitor EchoSense might need to prove in order to have a real place in the market, as Louise sees it. As you've heard already, and will hear repeatedly over the next month, this was an extraordinary meeting. These conversations should give you a flavor of the broader discussions we will offer in the next several episodes, starting with drug development on June 20th. And our review Friday is with Louise. Louise is now back in London. How are you?
Louise Campbell.
I'm very well, thank you very much. It's nice to be home. It's always nice to be away. It's nice to be with the liver community, but it's nice to be home with a cup of tea.
I missed the liver community this week, and I totally understand that.
Yes, we missed you.
Then in a little bit, I'm going to be speaking with Sven Franke, who will talk about Friday and Saturday, since he was part of the CPG committee, which I think is Friday's big story. And he was the chair of the late breakers, which were pretty remarkable, actually. The tirzepatide and efruxifermin presentations were exceptional. I believe, although they're both phase 2b studies, that we are truly turning a corner right now. And when you take the CPG recommendations together, it's really interesting. So, first of all, general impressions since we last spoke. You said you got to a couple of things on Friday, maybe.
I got to a couple of things on Friday for the first time.
We were texting during the CPG guidelines.
We were. Well, you and John and you and I. But I got the impression it was a really exciting meeting from everybody's standpoint. I think there were a few comments that different people had said about the fact that it started all about public health and the research content. And there was a couple of comments made to say that it was still all about p-values. And of course, a lot of public health research is often more qualitative rather than quantitative. And they thought that there might have been a bit more of that. And I'd heard that from several people. So it seemed to be a general consensus as we're moving into public health. They expected to see more qualitative rather than quantitative, and that particular focus on the academic p-value, the data, which I just thought was interesting because we obviously talk a lot about p-values here and the serious data, but I could understand where they were coming from. So it was interesting.
When you get into public health, right, you're fundamentally about consensus, opinion, and then legislators. And one of the things you learn about legislators is that they're all about data. You can't sell a feeling to a legislator because they're checking their polls. So you have to be able to show them in some way that's demonstrable, either that the policy decision is incontrovertible or that the people that matter are on the side of the decision that they want to make, one or the other. Take your pick. So I get that. What sessions did you make on Friday besides the CPG one that we were at that was so fascinating?
I listened to the nutrition one because I couldn't get in. With 5 minutes to go, they closed the door, which was really encouraging because lots of people were in that. And there was another 25 or 30 of us listening and watching online because it was a general discussion. And that was excellent. I went to the MASLD non-invasive posters that was presented by Jérôme Broussier, and we had heavyweights there. We had the Maya Thiel presentation from one of her colleagues. We had Alina Allen's colleague present. So we had a lot of heavyweights around this one, and I'll discuss a little bit more about one of the posters. Jean presented there. So this was a heavyweight session, and I did some of the earlier stuff on non-invasive technologies earlier in the room that Jérôme was presenting at, but so were some of the others. So I got to different things and dipped in and out of a lot. And then I was in the CPG session, which blew my mind. It's like the podcast has weaved its magic spell, and the leap of faith that I talk about and sometimes get shot down. We need evidence. But obesity is not going to stop driving. Diabetes isn't going to stop increasing. We need to take a leap of faith for liver disease. We did more than that yesterday with the CPG. So I was just so excited. So as we were talking to each other, I've never known anything like it.
I couldn't agree with you more, as you know. What's even more interesting, though, is to view that session in the context of what happened at the Late Breakers today.
Which I missed because I was flying back.
But the 2B on tirzepatide shows that, in fact, it has a significant impact on fibrosis, right? Maybe 25% in the placebo group on fibrosis, 50%, if I remember roughly. These are very rough numbers. I'm not looking at the paper. The paper published today in New England Journal of Medicine, so you can find it easily enough. And Lilly actually put out a press release on it on Friday, but it kind of jumped the gun, which surprised me. But it demonstrated that tirzepatide had a significant effect. And then it showed a really powerful 96-week effect for efruxifermin, which suggested, I think, pretty clearly that the concerns about wear-off effects on FGF21s are not a thing that matters, or not a thing that we're going to wind up being concerned about at the end of the day, right? And then second, and beyond that, if you put those up against CPG, which suggested that under certain circumstances, it may be appropriate to use drugs that aren't approved, or take into account drugs that aren't approved. So when they say you can't use tirzepatide for fibrosis, but you can use tirzepatide in diabetes and obesity, wink, wink, I think that's right. And one of the points that was made somewhere, I think this morning, is that you can only get tirzepatide paid for if you have diabetes. But even so, that's a large chunk of the MASLD population and the MASH population. So all things considered, not bad.
Certainly, I did not expect the CPG to go where it was. And somebody asked me this morning why I was still in shock. I have never known a guideline come out approving medications and naming the medication, resmetirom, to be used in the MASLD field in a region that the drug is actually not approved in yet. So that is going ahead of, normally we would wait, we would get a bit of data, we would do it, and then we would review it because we've already published guidelines. And then they just have to be adapted. Never have I seen a guideline in any form of hepatology, and I may have missed a lot, I assure you, but I have never known a guideline pre-approve a drug for use that's published in advance of the drug being approved for use. So that is a leap of faith, beyond a leap of faith. But I'm dead excited about it because they also commented on lots of other drugs. Pioglitazone may not be useful it can't be recommended in fibrosis, but you can use it in. So they were more comprehensive in the medications. They might not be used for a specific area of MASLD, but they could be used in this way. And I think that's excellent when you're not a non-hepatology specialist.
Yeah.
But remember, this is the Society of Endocrinology and Obesity coming together with hepatology. And the only thing I can think as a title for this is hepatometabolic health. Not liver disease, not obesity, not endocrinology, but this is more the overall health. Because where you look at the non-invasive list of cutoffs, they've nailed FibroScan at 248, which is the meta-analysis score. And remember, a lot of people now are not going to be biopsied.
Right.
So all of the grades for fibrosis normally that you put with FibroScan is when you've got a confirmed diagnosis.
Right. So
where I know somebody's got alcoholic-related liver disease, I use the cutoffs for alcoholic. And on the new ECOSENSE liver management platform, you can pull down each guideline to change the scores and to change the reports, for example. So they've allowed that leeway. But for meta-analysis for people with steatosis, that 248 is based on, I think, 4,500, nearly 5,000 liver biopsies of all cause. So I was interested to see that because a lot of the abstracts that I read throughout the conference use different cutoffs. And when we talk about MASLD and MASH, we tend to talk about cutoffs of over 280, some go into the 300s, but we have to put it in the context that we are definitely looking for somebody who's going to, on a biopsy, fulfill criteria for a clinical trial. But give me somebody with a cap of 249, 250, 260 now, in their early 20s, we can now start to manage them with this type of guideline, which offers the opportunity to get in early, prevent and manage lifestyle to prevent diabetes and obesity potentially, or slow it down. And that's where I come into now about, and have done for the last few years, as you know, about liver health is the issue and the comorbid situation, and liver disease is the end stage to start early. And this CPG really, really, for me, delivers that potential. Will we see that roll down to the international countries themselves? I'm sure the UK will look at it, and we certainly have to do our guidelines. They were written in 2015. But yeah, you couldn't have asked for anything more. In fact, I certainly wasn't expecting as much as it delivered. So well done to everybody in that.
So first of all, my sense is that that was a prod to the individual countries, right? Because not only is it a guideline, but it's a guideline from 3 societies, and they didn't talk about any drugs that aren't covered for diabetes. So that was my first thought. My second thought was wisdom morphs over time. And as often as I've quoted our friend Stephen as saying that liver is the canary in the coal mine, I think the way people are looking at this now, it's a little bit different. That in fact, metabolic disease is like the gray fog, and it's liver, and it's diabetes, and it's obesity. And while those are the diseases, the causes of death will be cardiovascular and extrahepatic cancers, obesogenic cancers, if you will. So the gray fog descends, and then eventually everything goes dark. And what liver has always been is the hardest element in the gray fog to define. Not necessarily the first sign, but the thing in the middle of that dark cloud that tells you that it's about to pour. And that's kind of how I took these guidelines and the organizations that put them together, because it was interesting when they did multi-organizations in the U.S., right? It was under the aegis of AGA, gastroenterologists, and included endocrinology, liver, primary care. This was a lot more metabolically focused, because if you think about the 3 major, if you will, I'm going to call them syndromes. I was taught a long time ago that the meaning of the word disease originally was all about pain, dis-ease, literally, uneasiness, right? Pain. And that, in fact, a lot of what we're calling diseases aren't really in terms of you don't die from them, per se. Nobody died from obesity, per se, or from diabetes, per se, or from, MASH, per se, until it progresses to cirrhosis or liver or HCC. So it's this gray mass of cloudy conditions that collectively comprise a dark cloud that cover the individual landscape. And then you're just waiting to figure out what the rain or the lightning or the thunder is going to look like and where it's going to land. Those are the causes of death: cardiovascular, stroke, obesogenic cancer. But these 3 organizations coming together this way is, I think, a really fantastic acknowledgement of what's got to be done. I agree with what you said about what they recommended was massive, but the group of people that they put together to do it, I also thought was different than the states and massive in its own way.
And their guidelines previously hadn't gone as far as, we've got to take the previous combined guidelines in the context that was several years ago, and where we've developed with liver disease, non-invasive technologies, biomarkers, and medications in that timeline has basically developed at speed. And I know it takes a lot of time to get trials to develop at speed at the end, and all of a sudden they come in. So I was, as you know, very happy about that. And
I know we were texting, I noticed.
But I think the overall conference, I got the sense that it was building to something. Everybody was excited. Everybody enjoyed it. So, yeah.
Alexander was certainly suggesting that the week before. I am immensely impressed with him. His energy, his enthusiasm, his vision, and his finesse, all that. He was massively enthusiastic the week before. And now he's always an enthusiastic guy, but I wondered if there wasn't something more behind it. And because I knew what was in the late breakers at that point in time, because we had gotten them, they were embargoed, but we'd gotten them because journalists, I knew that tirzepatide and fructofermin were going to show up. So I've been having conversation with people in the States, some of whom were clients and some of whom were friends, about what they're now calling the GGs and the GGGs are going to mean for Madrigal and really the mainstream oral drugs, right? Because the question now is there a pincer movement or not? The pincer movement would be people going on incretins of some form or another in the first place, or if they're really severe, going on the FGF21s. And then in the middle, you've got Resmetirom, you've got lanifibranor, you have all the THR betas as they show up. And those are kind of middle of the road drugs. Part of me says that squeezes the middle. And part of me says that as long as you have 60% GI side effects with tirzepatide and what the FGF21s are going to cost, that it's going to look more like what Stephen used to suspect would happen, which was oncology, right? Acute treatment and then maintenance. And on maintenance, everybody works their way to Madrigal because it's safe and they're not going to get worse. So when I look at the big picture, those are the 2 sweeps I see in terms of the drugs. Do you have any sense about which one of those it might be? I don't yet. I'm going to ask a lot of people that question.
I don't know. And I think my focus is more on the monitoring, the detection, rather than the sort of method of action and the type of drugs. We might move into the stage where we were with patients on hepatitis C. It's the right patient, right time, right drug. There are drugs, for example, that don't particularly lose weight. And in fact, one of them has a weight gain aspect. Now, I deal with lots of people who don't want to lose their curves. Lanifibranor them. They don't want to lose their curves. We don't need to put them on drugs that force the stereotypical weight loss. If they don't want to lose their curves, the potential is they can have a drug that may have a similar effect, but actually keep their body shape. So we get to the stage where we can individualize the drugs around what the patient's needs are and the way they feel. And I think having that portfolio of medications, everybody just assumes that we have to lose weight. We know these drugs can lose internal weight and not external weight. we did it with hepatitis, right time, right patient, right drug. A lot of it is about the patient. When is it suitable for them to go onto it? And we're going to know that with the fibrosis scores, but all of them having different actions and different ways, I think we can support a lot more. I think that's great, but I think it becomes part of the questioning. What drug would suit you best? Because we now have a portfolio. That would be a beautiful place to be for where we were when we started.
5 years from now, I think we'll be there.
Yeah, and 5 years from now, I think we will be there.
We've got a bunch of good 2Bs and 3s. Okay, so now let me jump over to devices, okay? Because at the same time that CAP is becoming more widely accepted and that EchoSense now says they can scan 96% of all people with BMIs over 30 accurately with the next generation product, which is what I'm seeing ads for or comments for. I'm hearing from other places comments that the holy grail is going to be in-office PDFF, that it's a better measure than CAP, and that if you can get in-office PDFF, that's the winner or superior. How do you feel about that? I don't know how I feel, and you know a lot more about this and think about it a lot more than I do. How do you feel about that?
I think you have to make sure the surrogate of MRI PDFF actually matches MRI PDFF. And I think we've moved beyond the stage where we're going to biopsy enough people on non-invasive devices, because we're not biopsying, to get the data that MRI PDFF have, or MRI, and FibroScan. There's over 4,800 publications, and that's before this meeting. So where do these other devices get that biopsy data from? Because we've moved beyond the biopsy.
Oh, and they're huge, and that's going to explode.
So surrogate of a surrogate, how do you prove it? And I think for Office at PDFF.
All right. So if I had a PDF, in-office PDFF device, I wouldn't even worry about biopsy. I would use the nail NIT logic. I would go past that, and I would simply try to match up to MRI PDFF.
But then you've got to match it, and you've got to prove it. And that takes a while. So I do think we'll get there, and that'll be great.
Well, so the question number one is that a commercially viable goal? So if you look at what's coming up right now to compete with the EchoSense, you get companies that say they can do better caps or easier caps, or not get a more reliable cap score, but fundamentally match the FibroScan score and do it in ways that are either economically or logistically favorable. And then you have companies coming up that are going to bet on in-office PDFF.
I suppose my question would be, from the scientific perspective, you might match the cap of EchoSense, but do you match the liver tissue that CAP matches for EchoSense? Those are two different questions. And one comes with biopsy data.
And I'm sure EchoSense will ask that question a lot.
Yeah, but that would be what Jean would ask me. Those are two different questions. One you might say, but you can't prove. I'm not the scientists behind this. For me, it's about, if you're going to use an MRI PDFF in an office, that's going to be with a physician. Mm-hmm. The VCTE with EchoSense, whether you look at a hepatoscope, whether you look at all of the other devices being brought in, the data isn't there yet. That doesn't mean it won't be there to say that it can be in the next CPGs, because of course they were very descriptive on VCTE or other blood-based biomarkers. No other device I saw in the guidelines that I've read, and they weren't discussed on the screen. That doesn't mean that won't develop, but the evidence at the moment isn't there. And I think we'll progress with the evidence, which will be good. But a PDFF machine, it will be a physician use, and it'll be in one office, probably because of cost. Rather like the initial FibroScan machines and the initial machines of any of these, they're quite expensive. Therefore, you tend to put them in the central hospital area so that people get. So I think it depends on how they're rolled out. I think the opportunities are there to get more evidence if we're going to be finding more patients. But I think it is going to come down to the evidence. Yes, I use FibroScan, but FibroScan was I've used since 2006, 2007, because it was the first and only. So I didn't have the luxury that we now have in this armory that is potentially come out. But it is all about the evidence, and that's the key.
So your point is an interesting one, which is what this might come down to in the end is whether A, regulatory agencies, and B, guideline organizations, and C, physicians will require a match back to liver tissue, or whether they will say, if you're sufficiently highly correlated with the non-biopsy tool that exists, we will accept that.
I don't, and I'm not making those decisions. I just operate the devices at the end of it.
I hear you.
And those may be totally different devices in 5, 10, 15 years. We don't know where we're going. But at the moment, I just use the one that I've used for nearly 18 years.
Okay.
So it's that, but that doesn't mean there's the evidence establishes, and I'm sure their machines will upgrade. And the new guided VCTE has lots of positive benefits. That's the development of technology. I use different technologies, but I more think about where can we use them for patient benefit? How can they be used? But I do lots of things, paracentesis clinics, things like that, where I've developed those pathways. It's just part of what nurses will do, venesection to help. We look at the way we can utilize technologies for liver at the coalface with patients and develop pathways to support our physician colleagues in delivering the best care to patients.
And with that, let's say goodbye to our audience for now. So welcome back to England.
It's nice to be back. But Milan was lovely. It was autumn temperatures in Perth, 28 in the day and 18 at night, which is what I left at Perth in the late autumn, early winter.
Oh, that's gorgeous. That's gorgeous. That's gorgeous. So it almost felt like you hadn't left.
Exactly.
Excellent. Okay.
And now back to Roger.
We hope you've enjoyed this recording. If you have any questions or comments about the content of this conversation or the entire episode, please put them in the review section of the page from which you downloaded this conversation, or send an email to questions@surfingmash.com. Until then, stay safe, surf on. We'll see you on the podcast. Bye-bye now.